Welcome to the Ecological Continuity Trust webinar series. A little bit of housekeeping before we start. If you can just keep yourselves muted during the seminar, and any questions that you have during the seminar, you can pop into the chat, which is just a little speech bubble in the bottom right-hand corner. At the end of the seminar, Ben will go through the questions and may well invite you to comment on your question as well. If you can also turn cameras off just to save on bandwidth, that would be very helpful. I'm very pleased to introduce Nina Overtoom, who is joining us from the University of York, where she's a PhD student. Nina studied her bachelor's degree in the University of Amsterdam, where she was particularly interested in soil organic carbon. And then she continued with an international master's at Ghent University and Aarhus University in Denmark, where she did her fieldwork in the Ecuadorian Andes, which sounds fantastic. Now she's a PhD student at the University of York, where she's looking at the effects of climate change on peatlands, with a particular interest in underlying processes. Nina, over to you. Do you want to share your slides?
Good afternoon, everyone. First of all, thank you to the ECT for the opportunity to speak today to you. My name is Nina Overtoom, and today I'll talk a little bit about the exciting research from the Fochno research site, which started in 2010 and is still ongoing. The main focus will be on what I did in the last 2 years, but also include some of the research which started way back in 2010. What I'll talk about is first a little bit of the history of Fochno. Then I'll talk about the setup of the climate change experiment and what we do. And then I'll go into some of the results from the long-term vegetation study, and then talk about some of the very recent results that we got from studying the greenhouse gas fluxes on site last year and this year at a summer drought. Why do we study peatlands? Well, peatlands are very large carbon stores. They store about a third of the world's soil carbon, but they cover a very small part of the Earth's surface, about 3%. Normally in peatlands, decomposition is slower than the accumulation. So slowly over time, you build up peat, which can reach up to several meters of just undecomposed plant material. And sphagnum species are an integral part of this ecosystem. They're keystone species. They maintain these wet conditions. They outcompete other species, create acidic conditions, and they form the majority of the peat as it decomposes very slowly. But these peatlands are vulnerable to climate change. So we get climate warming, we get more increased intensity and frequency of extreme events like droughts. And on the climate change, we can get vegetation shifts, and all these things can have effects on the carbon balance of these peatlands, because we don't quite understand how these combined effects, what their effects are, and that's why we study this. So this is Fochno. This is a picture that Justin gave me, and Fochno is a raised bog of around 7 square kilometers on the west coast of Wales. It's a very beautiful site. It's one of the few remaining near pristine lowland raised bogs in the UK. So it's quite unique. Fochno started forming around 6,000 years ago from a salt marsh, then transitioned into a fenland, a wet woodland. And then over time, the fen started to fill in with vegetation. And now the raised bog is formed predominantly by sphagnum mosses. And over the last 4,500 years, the bog is laid down up to 8 meters. So in the center of the dome, there's about 8 meters of peat, which sits on top of blue estuarine clay. In the 1800s and 1900s, parts of the bog were drained for agriculture and drained and dug up for domestic peat cutting for fuel. and therefore today the area of pristine bog is only a fraction of its original size. I mean, now it's about 7 square kilometers, like I said, but if you compare that to the 24 square kilometers it was in 1800s, but still it is there, and it's part of the Dyfi National Nature Reserve, and it's managed by Natural Resources Wales. The conservation of Fochno began in the 1950s when it was designated SSSI, and from then until the early 1980s, much of its conservation focused on acquisition by the state and preventing any further damage. And since the late 1980s, the long process of reversing over 200 years of damage to the bog started. And I'll go into some of the restoration work that has been done briefly, because the work focused on re-establishing favorable hydrological conditions, with an aim of getting the water table between 0 and 10 centimeters below the soil surface, reducing shrub encroachment, an area where it was drained before, controlling rhododendron, and using cattle and pony grazing in some of the other areas which where it was impossible to establish favorable hydrological conditions. So the restoration at Fochno, the work that's been undertaken to re-establish the favorable hydrological conditions over the last 35 years, sort of you can divide it into 4 main measures. So there's been damming of historic drainage ditches using peat from adjacent areas, which can cover a small area of about 0.5 meter to a much broader ditch, which require dams up to 50 meters wide. And many kilometers of ditches have been dammed using this technique. And then there's been peripheral pressure bunds, several kilometers of that have been installed. Use of recycled plastic piling to raise the water table in small ditches and old peat cuttings, and extensive areas have been dammed using this technique above approximately 250 hectares between 1997 and 2016. And the last measure is low contour bunding, as you can see on the right-hand side. And these bunds are about 25 centimeters high and created by repacking a linear trench to plug subsurface cracks. And this is followed by building a bund out of ongrotrophic beat sourced upslope of the bund. This holds back surface water, creating open pools behind the bund. That provides an opportunity for aquatic sphagnum species to establish. And between 2016 and 2023, around 45 kilometers of low contour bunding has been constructed, and the majority of this was funded as part of the Live Welsh Raised Bog project. Currently, the vegetation on site is dominated by sphagnum species, in particular Sphagnum pulchrum, but other species and plants that we find are whitebeak sedge, other sedges and grasses, some heather species, cotton grasses, the very beautiful coniferous sundews, liverworts, and it's also home to some rare species, including rosy marsh moth and bogbush cricket. And now on to the climate change experiment. So as I said before, because of the threat of climate change to peatlands, 15 years ago, scientists Richard Payne and James Rowson set up the climate change experiment at Fochno. And since then, many scientists, PhDs and students have been involved in this research. Work of Martin Kay included the effects of seawater flooding, greenhouse gas flux emissions. Luke Andrews focused on paleoecological record and matched that to current vegetation changes. So lots of research has been happening. And what attracts all these people to this site is that it's a very unique experiment. It tests warming using open top chambers, as you can see on the screen. And it combines that with summer drought. So what really sets this experiment apart from other experiments is that it tests the effect of a temporary summer drought. So over 3 weeks, the water table is lowered by pumping the water from the plots, and this allows testing of the effect of short-term summer drought, the recovery of this. And you can see that better here. So this is the climate change experiment where We test warming using these open top chambers. So we have a warming treatment, we have a drought treatment, and a control, and then in the warming and drought, so the combined treatment, they're combined, and then we repeat that in 3 blocks. So that's how it's been set up. That's how it's set up in 2010, and we're still using that. And then at the site, we measure things like air temperature, soil temperature, water table depth, and natural resources Wales. They have a weather station on the bog, which measures things like rainfall and temperature and radiation. So that's also really useful. So how do we do the summer drought? As I said, this is a little bit different from other drought experiments on peatlands. So there is a history of droughts on the site. So there was a drought applied in 2010, 2011, 2014, 2019, and then I did a drought last year. This is really where the ECT came in, because of the funds of the ECT and the Ramboll Worldwide Outdoor Trust, we're able to replace the pumping equipment, the monitoring equipment, and we're able to perform and apply this summer drought last year. So how did that work? Well, on the screen you can see the water table depth below the soil surface, and then this is across time. So the different colors indicate the different treatments, with control in gray, warming is orange, and then combined is red, and drought is in blue. And you see, as soon as the drought started on the 21st of August, the water table started to drop. So we achieved a drought, dropped to about 20 centimeters below the soil surface, and sort of maintained that over time, and then it came back up after we switched off the pumps on the 16th of September, and then the water table rose. And because I'll also show some greenhouse gas fluxes later today, I'll just point out when I measured them. So I measured them before the drought started, and then a couple of times during the drought, and then one time after. And then I went back this summer as well to measure the sort of recovery from the drought, but like a year later. So we have those data as well. And that's because in my research, I'm interested in the sort of short-term response to drought, but also the long-term legacy effects of repeated summer droughts, where we can ask questions such as, do peatlands become more resilient to drought or more vulnerable? And that's really relevant in light of climate change. So today, go into a little bit more of the research. My overall question is, what processes drive peatlands' response to climate change? And then today I'll talk about the first 2 questions. So how does vegetation community composition respond to long-term climate warming and repeated summer drought? So I'll focus on the vegetation changes. And then I'll go into the second question, what are the effects of climate warming and summer drought on greenhouse gas fluxes? And the third question we have to leave for now, because that focuses on the soil biogeochemistry and soil microbial community. And I'm still analyzing those samples in the lab, but maybe in another talk. So let's first look at the vegetation data. So every year, the researchers come back and measured the vegetation, started in 2010, and this has happened annually. And this is done using the pinpoint method at like set places within each experimental plot, where the number of touches per plant species is recorded, and this is then added up. So it gives you an idea of the abundance of the plant species in a particular plot. Let's go into some of the very exciting results, because it's a very cool data set. So when we measure the vegetation, there's about roughly 20 different species, and we group them into different functional groups as they have different functions in the ecosystem. So we group them into sedges and grasses, and then sundews, sphagnum, and shrubs. And on the screen are 2 of the groups which haven't changed. So on the x-axis, you can see the different years. So starting in 2010 and then going all the way to 2024. As the bog is dominated by sphagnum and by, at the moment, white-beak sedge. And it's something that has become more abundant on the bog, but it's not something that's become more abundant as a result of our treatment, but just in general across the site. And sundews haven't responded either to our treatment. But shrubs have. And Luke Andrews already found this when analyzing the data up to 2020, that warming was found to have a significant effect. So when we compare the different treatments, warming is in orange-yellow. You can see that the warming treatment, the abundance of shrub really increased in the last couple of years. It was already found in 2020, and this has just increased further. And this is not something we just see across Fochno. I mean, this is observed in other northern peatlands where we're seeing an increase in vascular plants, in ericaceous shrubs. And it's really relevant to know what happens then further onto the ecosystem, what the effects are on carbon cycling. So these are the shrubs. And then the last group, I should just say that this increase was driven by 2 species. So Erica tetralix, we can see, has increased on the warming, and Collina vulgaris has increased on the warming. And then the last group are sphagnum species. So sphagnum species, like I said, keystone species for peatlands. We see that up to 2020, there wasn't a big change in the sphagnum's abundance. But then after 2020, we start to see this drop in the sphagnum species, sphagnum abundance going down. And if we combine this with the increase in shrubs that we're seeing and a sort of a decrease in sphagnum, we start to wonder, is this a potential system shift? What are the effects on carbon cycling? As is very relevant to research further. And that brings me nicely on to the second question. So what are the effects of climate warming and summer drought on greenhouse gas fluxes? And this will use the data from collected in 2024 and 2025. Just first to give you a little bit on the methods, and then we'll go into some of the results. So the experimental setup on the screen, you can see the different treatments, so the control plots, the warming plots, drought, and then warming and drought. And then per plot, measured the greenhouse gas fluxes both on an area dominated by sphagnum and an area dominated by heather. And the heather was, we saw this increase in shrubs, and that was driven by Collina vulgaris, common heather, and Erica tetralix, cross-leaved heath. So we measured the greenhouse gas fluxes on areas dominated by common heather and cross-leaved heath. And this is done so on 2 vegetation types in each treatment plot. When we do greenhouse gas flux measurements, we use a greenhouse gas analyzer, and we used a see-through chamber, which was placed on a collar in the soil, which provides an airtight seal. And then we use a Los Gatos ultra-portable analyzer, which measures the gases every second. First, we air out the chamber, and then I do a light measurement. That means under light conditions, so there's both photosynthesis and ecosystem respiration, which gives us a measure of net ecosystem exchange. And then in the middle picture, you can see that I air the chamber out, and then that's followed by a dark measurement, and dark measurement, which means that we exclude the light, so there's no photosynthesis, so we just measure ecosystem respiration. So let's go into some of the results. I'll first present the net ecosystem exchange results for CO2, then the ecosystem respiration results, and then the methane fluxes. So let's look at CO2 net ecosystem exchange. So the question here is, how is CO2 net ecosystem exchange affected by climate warming and summer drought? So on the screen is a lot to take in, but I'll talk you through it. So we see CO2 net ecosystem exchange over time. So there's different dates that are measured. Just note that the dotted line is when the summer drought started and when it ended. And then there is a jump here in time from 2024 to the summer of 2025, just to note the jump in the x-axis there. And then on the y-axis, we see CO2. And that's the first thing. And we notice they're negative. So on the top here, I should just say sphagnum, and then on the bottom here, we have header. Colors correspond to the different treatments, which you can see at the bottom here. And the first thing to note is that all the fluxes are below zero. And that means that there is an uptake under light conditions of CO2. So that's good. It's a sink for CO2. And the next thing that we can start to look at are the treatment effects. So on some of the days, not all of the days, but on some of the days, drought seems to reduce the sink for CO2. So that means that when we compare the gray boxes to the blue boxes and also to the combined, because combined also has drought applied, during the drought, we see that the sink for CO2 is smaller, it's closer to zero. But there's a lot of variation between different days. So again, we see it for some of the days on the header, but not on all of the days. What is much clearer is that in general, sphagnum has a smaller sink for CO2 compared to header, because header is further away from zero compared to sphagnum. That becomes a bit more clear when we look at this graph. So here we see the effect of the vegetation type on the CO2 net ecosystem exchange, with the blank boxes are sphagnum and the striped boxes are header. Then we have control, we have drought, warming and drought. And what we see is that this is averaged across all the different dates. And we see that every time the striped boxes, so the header boxes, the net ecosystem exchange is more negative. So it's a larger sink for CO2 on the header. Then what happens with respiration? So now showing the results for the dark measurements, so no light, no photosynthesis, ecosystem respiration. And we see here that, again, we've got sphagnum on the top, header on the bottom. And we see that the fluxes are positive. That means that there's a loss of CO2 from the soil and the plants into the atmosphere, so a positive flux. What are the effects of the different treatments? And some of the days, not all of the days, but some of the days, warming seems to increase respiration. So when we compare the orange and red boxes, which is where warming is applied, to the control and drought boxes, we see that the respiration is higher. And that also happens sometimes on the sphagnum. But again, what's more noticeable is the difference in vegetation. So we look at respiration here across all the different dates, compare sphagnum to header. Again, striped is header and the blank boxes are sphagnum. In all the different treatments, the respiration, ecosystem respiration, is higher on the header compared to sphagnum. So when we tie this into the changes in the vegetation community composition that we saw, as we get more header becomes a larger part of the mix of the vegetation. The CO2 fluxes, or both net ecosystem exchange, but also respiration, these will increase in magnitude. And what happens to methane emissions? So what are the effects of climate change on methane fluxes? This is a very similar plot, but now with methane. And when we look at the y-axis here, we can see that there's methane and that there's positive fluxes for all the different days. So we have a release of methane from the system. And then when it came to methane, we noticed a very interesting interaction between the dominant vegetation type and the response to the different treatments. So on the sphagnum top graphs, we see that drought on some of the days seem to reduce the methane fluxes. So we compare the blue and the red, which is where drought is applied, to the non-droughted, that on quite a lot of the days that the amount of methane that's being released reduces when you have a drought. Now that makes sense, because normally there's anaerobic conditions, but when there's drought, there's more oxygen in the soil, so less release of methane. But we don't see the same on the header. So there's a different response to the climate change treatment depending on the vegetation type that you want. The effect we do see on the header is that warming seems to increase methane emissions on most of the day. So we compare the control and the drought plots to the warming plots, and we see that the warming plots have a higher methane release. Now compare that across all the different dates. We see that drought reduces methane emissions on the sphagnum, but not on the header. However, methane emissions are already lower on the header, on the control conditions. And then warming has a far larger effect if you move from control to warming conditions than if you do that same jump, comparing control to warming on the sphagnum. So a really interesting interaction going on. So it's quite a lot of data that I showed you. So what can we conclude from this? First of all, it's a very unique site, Fochno, and it allows us to study the response of vegetation community composition and greenhouse gas fluxes at a plant level under a long-term climate change experiment of both warming and episodic summer drought. And then to go back to the research questions, how does vegetation community composition respond to long-term climate warming and repeated summer drought? Warming increases ericaceous shrubs, and since 2020, it also seems to decrease the abundance of sphagnum species. And this really nicely ties into the ECT because we only saw this shift after a decade. So there's a real need to do long-term research to have these long-term sites. And then for the second research question, what are the effects of climate warming and summer drought on greenhouse gas fluxes? Across vegetation types, on some of the days we saw that the CO2 sink was reduced during drought and that ecosystem respiration increased on the warming, but more pronounced were the differences between sphagnum and header. So header has a larger CO2 sink and higher respiration rates. Header also has methane emissions, which respond far more to warming compared to sphagnum areas, but sphagnum, the methane emissions decrease during drought, and we don't have that same effect on the header. So we can conclude from this that if we see a shift in the vegetation community composition with more header, this will both increase CO2 respiration and the sink for CO2. And for methane fluxes, this might shift. This shift could reduce the response to drought conditions, but overall increase methane release on the warmer temperatures. And in my PhD, I'm continuing this work. We're now looking into the soil samples that were collected at the same time to look into the soil processes and the microbial community associated with these vegetation shifts and in relation to carbon cycling. I just want to thank you all for listening to me today, and I want to thank everyone who has contributed to this project and to the research at Fochno for the last 15 years. It truly is a unique site. Thank you.